Hello and welcome to another episode of the Prof series. For this episode, we'll be covering a paper by Dr. Barry Micallef. He's a researcher in greenhouse vegetable production and vegetable production in general, specifically around its physiology and genetics. He teaches 1/3 of the plant and animal genetics course and the vegetable production course. For this episode, we'll be talking about a paper by Dr. Barry Micallef regarding photosynthesis efficiency in tomatoes. Let's get started. Sugar is one of the primary products of photosynthesis in plants. Experiments in CO2 exchange with plants have found that the efficiency of which CO2 can be absorbed by the plant is one of the primary limiting factors within photosynthesis. This limitation occurs primarily in high CO2 levels in certain instances. So it's effectively a hard limit on what each plant species can absorb in terms of CO2 before it begins harming the plant itself. The question is, how do we deal with these limitations? Well, there's been extensive research in the genetics behind sugar synthesis in different organisms, specifically plants, and various tomato strains have been genetically engineered to exhibit different forms of this gene. So these genetically engineered lines were subject to different environments and different CO2 levels. The CO2 concentration fell under two treatment options, 35 Pascal of CO2 or 65 Pascal of CO2. And to maximize the amount of uniformity of the treatment option, they're grown in specialized growth chambers where CO2 was applied at a very even rate. They're all subject to the same light levels of a 500 micromole lamp at a uniform temperature of 26 degrees Celsius for a 16-hour day and 18 degrees Celsius for the night period. Irrigation was done through drip irrigation using a pre-made fertilizer mix that's applied at the same rate for all individual plants. Everything was made almost completely uniform. The only thing that differed between populations is the CO2 treatment. And here the results found something interesting. After 40 days after seeding, sugar production within the plant between all lines did not differ between each other in both experimental groups. So 65 pascals of CO2 and 35 pascals of CO2 did not make a lick of difference after 40 days after seeding. The genetically engineered populations of tomato, however, began languishing in the 65 pascals of CO2 system 50 days after seeding. Genetically engineered lines and control lines tested did not have different rates of photosynthesis, however. Based on the study, the maximum amount of CO2 absorbed by the genetically engineered line was 30 micromoles of CO2, 5 micromoles below what's in the atmosphere. The rate of photosynthesis for one of the genetically engineered lines was 33 micromoles of CO2, 27 micromoles of CO2 for the untransformed line, and 21 for a third genetically engineered line. The ability for the plants to fruit, however, was improved in both the 35 Pascal system and the 65 Pascal system for CO2. Overall, the GMO varieties produced more fruit in this artificial system than did the conventional varieties. Overall, the sugar production activities overall were far greater for the genetically engineered varieties than the control varieties. A similar phenomenon happened in our experiments using spinach. However, the results also found that increased sugar production capacity did not increase the rate of photosynthesis, but producing varieties in higher CO2 levels did increase the rate of carbon acquisition in those transformed lines. So with these genetically engineered varieties, they do not become insensitive to CO2 at higher levels, unlike the natural varieties. This demonstrates that the rate of which end products are produced and the capacity to produce those products are a key limiting factor within photosynthesis, and a key limiting factor to tolerate, an even greater factor in CO2 acquisition for use in making sugars. That about covers everything. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.